Welcome to our video lecture on DNA and RNA and DNA replication. Our essential understandings for today, we are going to look at the structure of DNA and how it allows for efficient storage of genetic information. And then we're going to talk about how we can copy that DNA information in DNA replication. Our objectives for today, we're going to talk about the Hershey-Chase investigation. We're going to compare and contrast DNA and RNA structure. We're going to look at the antiparallel structure of double-stranded DNA. We're going to look at the contributions of Franklin, Watson, and Crick to our understanding of what DNA looks like. We're going to talk about the Meselson and Stahl investigation into semi-conservative DNA replication. We're going to look at a bunch of enzymes that are involved in DNA replication. And we're going to wrap up with a look at PCR. So this Hershey-Chase investigation, we didn't always know that DNA was the molecule of heredity. There were a lot of theories that protein was actually the molecule of heredity, just because there was so much more variation to those proteins. So this pair, Martha Chase and Alfred Hershey, they put together this investigation where they took bacteriophages. Bacteriophages are viruses that infect bacteria, viruses that make bacteria sick. Some of those viruses, bacteriophages, had radioactive sulfur in their proteins, and some of the phages had radioactive phosphorus in their DNA. We know that sulfur is only in proteins and phosphorus is only in the DNA, so it was easy to keep those two separate. The experiment then had these bacteriophages infect the bacteria, and if the protein was the molecule of heredity, then the radioactive sulfur would have ended up inside the bacteria cell to make it sick. If the DNA with the phosphorus was the molecule of heredity, then that phosphorus would have ended up inside the bacteria cell. So those phages infected, they did a wash to kind of get the virus coats off of the bacteria cells, and they centrifuge, which is a super cool spinny thing that separates things by density, centrifuge to separate the bacteria cells from the viral protein coats, and found that phosphorus, that radioactive phosphorus, was inside the cells. There was no radioactive sulfur inside the bacteria cells, which allowed us to understand that that DNA was what got into the bacteria cells to make them sick. It wasn't the protein. So Hershey and Chase determined that DNA is the molecule of heredity. So let's look at the structure of DNA and RNA. Some things that they have in common. Both of them have a sugar. It's a 5-carbon sugar. Both of them have phosphate groups, and both of them have nitrogenous bases. So everybody's got phosphate groups. Everybody's got some kind of 5-carbon sugar. We call it a pentose because it has 5 penta sugars. And then there's that nitrogenous base. Some differences, they're not the same sugars, right? So in RNA we have ribose, and then in DNA we have deoxyribose. And the difference literally is we lost an oxygen. So OH here on RNA, just the H here on DNA. Nitrogenous bases are also a little bit different. In RNA, we have adenine, cytosine, guanine, and uracil, whereas in DNA, we have adenine, cytosine, guanine, and thymine. Some other differences: DNA is double-stranded. DNA is double-stranded, and we have complementary base pairs being held together in hydrogen bonds. Those complementary base pairs are adenine and thymine, cytosine, and guanine. Our RNA is single-stranded most of the time. And again, we have that ribose sugar versus the deoxyribose sugar, but those nucleotides still have phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base. So now we're going to focus really just on DNA. DNA is a double helix. We have 2 strands of DNA. They're held together by hydrogen bonds. Those hydrogen bonds form between complementary base pairs. We have adenine and thymine, cytosine and guanine. Those are our complementary pairs of bases. We're going to focus here a little bit on this idea of antiparallel strands. Remember that in biology, we really love to count our carbons. And so we're going to count some carbons in our sugars, so those ribose sugars. So we have carbon number 1, carbon number 2, carbon number 3, carbon number 4, and there's also another carbon right here, carbon number 5. How to remember this? Carbon number 5 is always stuck to the phosphate group. 5-phosphate. They sound, well, they both start with that F sound. 5 and phosphate. And then carbon number 1 is on that nitrogenous base. Carbon number 3 is where the next nucleotide will be bound. So 1, 2, 3, 4, 5. Notice that the last carbon on this side is a 5 carbon, carbon number 5. Sometimes we call it 5' carbon. On the opposite side, we actually end with carbon number 3, 3' side. And when these guys line up together, they are antiparallel. This guy's going to run 3 to 5. This guy's going 3 to 5 this way. And so we line up 5s and 3s on opposite ends. We're going to look at that one more time, zoomed in just a little bit more. So again, here's our 5-carbon sugar, deoxyribose. We're going to label our carbons. Carbon number 1 is stuck to the nitrogenous base. Carbon number 2, carbon number 3, carbon number 4, carbon number 5. Carbon number 5 is where we find that phosphate group. Because carbon number 5 is the last carbon in this chain, we call this the 5' end of this DNA strand. But on the opposite side, down here, we have carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5 in the phosphate group. This is our 5' end. They are not lined up. So we go 5 and 3. We go 5 and 3. So we call this antiparallel because they run in opposite directions. These guys run in opposite directions. Good? Good. All right, so this idea of complementary base pairing, how do we even know that adenine and thymine go together and cytosine and guanine go together? This amazing guy, Edwin Chargaff, so he did an investigation where he literally took different kinds of DNA from thymus and the spleen and the liver and the sperm of different animals, and then compared quantities of adenine, thymine, cytosine, and guanine. And he found that the numbers were pretty similar, adenine and thymine, guanine and cytosine. Not exactly the same, but pretty similar. Adenine and thymine, cytosine and guanine. And that was true across all the different samples that he looked at. And so he deduced that adenine and thymine must pair up together, cytosine and guanine must pair up together. How do we know the structure of the DNA? Well, this is all about Rosalind Franklin and then those guys, Watson and Crick. So, Rosalind Franklin was an X-ray crystallographer. Her job was to X-ray things that she could turn into crystals, and then the way that the electrons would bounce off those crystals would make patterns in pictures, and people could deduce the shapes of molecules from those electrons bouncing around off of those crystals. We call that diffraction pattern, and that allowed us to build our atomic models. The job of Rosalind Franklin was to take pictures of stuff. So this is Rosalind Franklin. She was not well liked in her laboratory. The men that she worked with found her to be quite brash. They did not think that she was ladylike at all. And so she was not well respected, despite being an amazing scientist, because she just wasn't demure enough for the people. Rosalind Franklin took this picture. This is photograph 51. And another guy named Maurice Wilkins literally stole it from her lab and gave it to these guys, Watson and Crick. Watson and Crick took her stolen picture and figured out, based on that diffraction pattern, that DNA had to be this helical shape. And they got a Nobel Prize for that, and Rosalind Franklin didn't. But there's a lot of talk about Rosalind Franklin now, so we greatly admire her contribution to our understanding of DNA double helix structure. Watson and Crick also did a lot of good work with that photograph, that stolen photograph, to figure out that DNA is this double helix. So Watson, Crick, and Rosalind Franklin are credited with our understanding of DNA is a double helical shape. So now we're going to focus on this idea of DNA replication, which is amazing and very, very detail-oriented. So be ready for lots and lots of words. We will practice these words in class too, so don't panic too much about all the vocab. But DNA replication is semi-conservative. This idea of semi-conservative was figured out by an investigation from Rosalind and Stahl. And it's super important to ensure that our genetic information is accurately copied. So this idea that DNA replication is semi-conservative was figured out by these guys, Rosalind and Stahl. And they did an investigation where they had DNA in bacteria grown in with food that has an isotope of nitrogen called nitrogen-15. This is extra heavy nitrogen. And then they switched the bacteria to food that had nitrogen-14, assuming that the bacteria would use this new nitrogen to make the new DNA. So we're going to start off with parent DNA, and of course all the parent DNA is made from that original food source that was nitrogen-15. So all these three different models are going to start with nitrogen-15, that heavier version of nitrogen in the DNA. If DNA replication is conservative, what will happen is that nitrogen-15 piece of DNA will stay exactly the same. It will be conserved. And our new copy of DNA will be made from the new nitrogen source, that nitrogen-14. So we'll have one piece that's all nitrogen-15 and one piece that's all nitrogen-14. And then we'll have another generation. We have that parent DNA will stay exactly the same, but now this is also parent DNA. It'll stay exactly the same. New pieces of DNA will be made from that new food source that's nitrogen-14 only. Nitrogen-14 only. And so then at the end of the second generation, we should have 1/4 DNA that's all nitrogen-15, and 3/4 of the DNA is all nitrogen-14. But if DNA replication is semi-conservative, we'll start with this nitrogen-15 parent DNA. We're going to switch the food source to nitrogen-14, and semi-conservative replication, the idea is that this double strand separates into 2 single strands, and then we add new DNA to each of those old strands, which is why it's semi-conservative. Half of the DNA is saved and half of it is new. If DNA replication is semi-conservative, then after our first generation, each piece of DNA will be half nitrogen-15 and half nitrogen-14. Half nitrogen-15 and half nitrogen-14 will have these hybrid versions of the DNA. And then after our second generation, these guys will separate again, and we'll add new pieces of DNA in that replication process when we copy the DNA. And so we should have one piece that's half and half, a piece that's all new, another piece that's half and half, and a piece that's all new. So we would have half our DNA as a hybrid of 14-15, and half of our DNA will be all new, all 14. If the last model were true, this dispersive idea is that the DNA would just break up into lots of little fragments, and then our new DNA would come in, also in lots of little fragments, and then they'd get bound together to make that new DNA. So it breaks apart and then comes back together. If this were the model, then everything should be a hybrid version of 15 and 14. And then these guys would all break up, and we would still have hybrid versions of 15 and 14. When they did this and collected their data, and we're going to look at that data in class, they found that DNA replication was, in fact, semi-conservative. And that semi-conservative replication is controlled by enzymes. So many enzymes. So this is the list that we're going to look at. We're going to talk about each one of them on these next couple slides. Each enzyme gets its own slide. First up is helicase. So helicase is an enzyme that is going to unwind, sometimes we say unzip, the double helix of the DNA. We need to get those strands of DNA separate in order to be able to copy the sequence of nucleotides that's in them. So helicase basically just breaks the hydrogen bonds that hold those complementary base pairs together. So helicase is here, just breaking up those hydrogen bonds that hold the double helix together. Topoisomerase, which is also called gyrase, so either one of those is fine, gyrase, topoisomerase, is going to relax the supercoiling. You can imagine that if you had a piece of twine or embroidery floss and you just started to pull the edges apart and you pulled really hard to try to separate those strands, that you're going to end up with a big knot down here. Same thing's going to happen with our DNA. It's going to get all knotted up if we just pull it apart super fast. So we have this enzyme topoisomerase, also called gyrase, and its job is simply to relax the supercoiling. And it does that by actually breaking the DNA and then putting it back together, breaking it to get rid of the tangles and then putting it back together so that replication can happen appropriately. So it's going to relax that supercoiling so that we don't get a bunch of knots in our DNA. DNA primase. This is a crazy sauce idea. So DNA is a little bit high maintenance. DNA cannot start replicating. DNA cannot be at the beginning of a line of new DNA. So we actually start with a little bit of RNA, which is far less high maintenance. So this enzyme DNA primase is actually going to put down a little piece of RNA. So 10 to 15 bases of RNA just to get things started. So this RNA primer, this is all going to be made of RNA, not DNA, for just a little bit. Once we have that RNA starting our strand of DNA, then we can start attaching DNA to the end of that little chunk of RNA. And the enzyme that does this is DNA polymerase 3. So polymerase 3 is going to attach DNA to the end of that RNA primer in a 5' to 3' direction. So remember how we count those carbons, 5' to 3', our 5' end, our 3' end. The 5' end of the new nucleotide is going to get stuck to the 3' carbon of the old or the previous nucleotide. So if this is where I am adding my, here's my polymerase, and I'm adding DNA in this direction. So here is my previous piece of DNA. It's going to be the 3' end of this DNA. I'm going to have my new nucleotide is coming in. It's got its phosphate group and its sugar and its nitrogenous base. That is not a very good picture. Let's pretend. So the phosphate 5' end is going to get stuck to the 3' end of that previous nucleotide. So we're always going to add the new 5 to the previous 3. And the reason is this, that phosphate group here, it actually has more than one phosphate group when it's separate from the DNA. And just like in ATP, where we have those phosphate groups that we can chunk off in order to put energy, transfer energy around the cell, same thing's going to happen here. So we've got all this energy in these extra phosphate groups. The polymerase enzyme is going to chop those phosphate groups off and use that energy to stick that last phosphate group onto the growing strand of DNA. So DNA replication happens in a 5' to 3' direction. 5' of the new nucleotide is going to stick to 3' of the previous nucleotide. So here's another look at that. Here's my new strand, my growing strand of DNA. Carbon number 1, 2, 3, 4, 5 is up here with the phosphate group. This is an oxygen. Here's my new piece of our new nucleotide. Carbon number 1, 2, 3, 4, 5 is up here by the phosphate group. And when we start, this phosphate group actually has more phosphates, and there's lots of high energy bound in those phosphate groups. And so that polymerase enzyme is going to chunk those phosphate groups off and use that energy to bind this 5' phosphate group to that 3' end of the growing strand. Good? Good. So we're going to build in a 5' to 3' direction. 5' to 3' direction. All right. So what happens because of this polymerase can only go 5' to 3' direction is we end up with DNA replication happening in separate directions on the 2 strands of DNA. Remember that our DNA is antiparallel. So this guy goes 5' to 3', and then this guy goes 3' to 5'. DNA polymerase can only go in one direction. So this guy, polymerase, helicase, polymerase, they're going in this direction. This polymerase can follow the helicase. We're just going to add more nucleotides as the helicase opens up that DNA. And we can move in the direction toward what we call, this guy right here is the replication fork. This is amazing and wonderful, and it can go super fast, and we call it the leading strand because it can go so fast. The other guy has to go that direction. So what happens is the DNA is going to get opened up by helicase. Polymerase is going to come in and work in that direction. And then another polymerase is going to come in and work in that direction. Then another polymerase will come in and work in that direction because we can only go 5' to 3'. Each one of these little pieces that forms on what we call the lagging strand, because it takes a little bit longer, each one of these little pieces that gets formed is known as an Okazaki fragment. Okazaki fragments form on the lagging strand of the DNA as it's getting replicated because polymerase can only go in a 5' to 3' direction. And on the lagging strand, polymerase has to work in the opposite direction of helicase. And so it takes a little bit longer, and we have these little fragments. Here's another image kind of showing that idea. So we've got our replication fork is here. Helicase is opening up that DNA. So here's our strands. Here's our original strands, and these are the new strands that we're building. On the leading strand, we can just follow that helicase and just keep copying. No need to stop. We can just keep going. On the lagging strand, we're going to go a little bit, and then helicase will open up more, and then we're going to jump back and go this way. And then we're going to jump back and go this way. And then we're going to jump back. We're going to jump back and go this way. And then helicase will open up a little bit more, and it will have to come back here and go this way. And each one of those pieces is called an Okazaki fragment. And it's all because polymerase can only go in a 5' to 3' direction. All right, so we put down those primers at the start of the leading strand and also at the beginning of every single Okazaki fragment. Every single Okazaki fragment on the lagging strand needs primase to start with a little bit of primer that's made of RNA. So we've got this guy, exonuclease, and its job is to literally take out the RNA primers. So exonuclease is going to come in and remove those RNA primers. And then this other polymerase, polymerase 1, is going to come in and fill those gaps with DNA instead of RNA. So we're going to take out those RNA primers and then fill in that little gap right there with DNA. But I still have these little gaps between my Okazaki fragments. And so we have one more enzyme that comes in, DNA ligase. And ligase is going to just glue together those Okazaki fragments. It'll glue together those Okazaki fragments on the lagging strand. Good? All right. So We in technology have been able to steal the idea of DNA replication, and we call this process PCR, polymerase chain reaction. PCR, polymerase chain reaction. And what we do is we basically hijack the process of DNA replication. So we start with our double-stranded piece of DNA. We're going to heat it up, which is going to cause the hydrogen bonds between the double strands of DNA to break apart. We're going to have denaturation. So now I have single-stranded DNA. I have some primers that I made in the lab on a sequence that I wanted to get copied, and I'm going to allow those primers to anneal or stick to our template DNA. And then we're going to throw some polymerase in there, and the polymerase is going to add in a 5' to 3' direction DNA to copy the sequence of DNA that I want to get copied. What's kind of cool is that I'm going to denature at about 95 degrees Celsius. So we heat up that DNA, and basically those hydrogen bonds just kind of melt. And then I cool it off a little bit, and that allows my primers to stick to the DNA. And then I warm it back up just a little bit, and that induces my polymerase to start copying the DNA. This does not work with our human polymerases because this is way too hot. Our human body temperature is 37 degrees Celsius. 72, our enzymes are very, very broken. They are very denatured along with our DNA. So what we do is we steal this polymerase from Thermus aquaticus, a bacteria called Thermus aquaticus. Can you guess where Thermus aquaticus lives? It's a hot water bacteria. He loves the heat. And so we steal its polymerase 1 and 3, and that will do replication in the lab at crazy high temperatures without getting denatured. The most amazing thing is that we can double the quantity of DNA in our sample just about every 2 minutes. Just about every 2 minutes, we can double and then double and then double our DNA because this amazing thing, Taq polymerase, Thermus aquaticus, TAC. Good? Good. All right, my friends, we did it in under half an hour. I'm so proud of us. Let's see if we met our objectives. Did we talk about Hershey Chase? Yes, we did. This was the one with the viruses that are called bacteriophages, and there was some radioactive sulfur and some radioactive phosphorus. The phosphorus in the DNA is what actually got into the bacteria to infect them, and so we know that DNA is the molecule of heredity. We compared and contrasted DNA and RNA. They have some things in common. They both have sugars, they both have nitrogenous bases, they both have phosphate groups, but then there are also some differences. The sugars are different, deoxyribose versus ribose, double-stranded versus single-stranded, uracil and thymine. We talked about that antiparallel structure of DNA. So our DNA is going to be 5' on one end and 3' on the other, 5' and 3'. We talked about Rosalind Franklin's X-ray crystallography photograph and how it got stolen, given to Watson and Crick, and this is how we know that DNA is a double helix. We talked about Meselson and Stahl just a little bit. They used the different versions of nitrogen, those isotopes of nitrogen 15 and 14, and figured out that replication is semi-conservative. We looked at those different enzymes. We talked about helicase and topoisomerase. We looked at primase and polymerase 3. Whoa, I can spell. We looked at exonuclease and polymerase 1, and then we talked about ligase because replication is cool. It needs a lot of enzymes. We're going to practice these in class. So if you're like, yep, nope, I definitely don't remember all those, don't even worry about it. And then we talked super quick about PCR, polymerase chain reaction. When we get to our unit on genetics, we're going to do this in class for reals with your own DNA. Well done today, my friends. And again, we did it under half an hour.